This one. Sorry, just confirm that you can see the presentation, please.
Yes, we can.
Yes.
Brilliant. Thanks. Okay, so thanks for the introduction, Colette. I'll correct you. I think I'm not quite an expert just yet, but I'm trying to get there. So welcome, everyone. As Colette mentioned, my name is Pieter, and I'll be doing this installment of the Next Generation Sequencing Webinar Series from Separations, but focusing on our Illumina technologies. And as she also mentioned, today's title is to realize the full potential of the NextSeq 1 and 2000. Although I think it's a very big title to try and live up to. I'll mention it throughout the presentation as well, but I think Illumina themselves are still discovering the true potential of these instruments. And my sincere apologies to everyone. I am just the sales monkey actually within the team that someone has deemed worthy to present the session. So hopefully I'll bring a bit more personality to the presentations, but maybe a bit less technical savvy in some of the previous talks we've had. Just to jump into it then, a recap from what Maria has told us in some of her previous talks, but the mission from Illumina's side is to improve human health by unlocking the power of the genome. And I truly believe that the NextSeq 1 and 2000 instruments are going to play a major role towards achieving that goal moving forwards. And to jump straight into the instrumentation side of it, for those who aren't really familiar with Illumina's lineup, taken here are just a few of the instruments. The iSeq 100 on the far left is the little benchtop entry-level sequencer from Illumina. And at the far right, we have the behemoth, the NovaSeq 6000, which is really a production scale sequencing system. The NextSeq 1 and 2000 instruments slot in towards the upper end of that scale, definitely. They're about 2 steps behind the NovaSeq. There is nothing 1 step behind a NovaSeq. It's just that impressive of a system. But it replaces the NextSeq 1 and 2000 now, replaced the NextSeq 550 from the previous generation, but also somewhat exceed its capacity. And just to clear up any confusion before it happens, the NextSeq 1 and 2000 instruments are virtually the same instrument. So it's that same box pictured on the screen. You'll see sometimes the color band in the middle will change color, but it's the same box for both instruments. The major difference is that the NextSeq 2000 has an additional internal module, which gives it access to the P3 reagent set. All these terms will be clarified in depth a bit later on, but suffice it to say for now, the two both occupy the space above the MySeq, but below the NovaSeq, and they're virtually the same instrument, barring that capacity. So what went into making the NextSeq 2000? It's the newest instrument launched from Illumina, the combo of the 1 and the 2000, and it was redesigned from the ground up to offer really a higher throughput than we'd seen before in a benchtop instrument, but also a lower cost. That's compared to almost any other technology barring the NovaSeq. So it allows for higher throughput applications than even the 550 would have allowed for. It allows the user to expand their operations by making use of this higher throughput of data at a lower cost. Similarly, that now grants scalability for deeper and larger studies. And what's really exciting that I'll spend quite a few slides explaining is their onboard informatics that they've included in these instruments, starring DRAGEN. So overall, really a powerhouse instrument that I feel is best aimed at a current sequencing user who wants to either decrease their sequencing costs or just increase their throughput and gain access to higher throughput applications. So the NextSeq 2000 and 1000 instruments feature over 75 breakthrough innovations. The highlighted ones there, first off, the chemistry was changed. So as opposed to the old 4-die chemistries on the previous systems, the NextSeq 1 and 2000 use a 2-channel sequencing by synthesis chemistry. This reduces the cost of the reagents as well as the optics, and also increases the speed with which the system can generate sequence data. But don't panic, it's still compatible with all the same library preparation modalities that the previous Illumina instruments have used as well. The next would be the super resolution optics that they developed, which allow for a really, really intense image of the flow cell so that you can get a really high density of reads from the flow cell, which ties in with the next development, the ultra high density patterned flow cells. So previous technologies from Illumina had the flow cell, which was a glass plate, and a random assortment of adapters across it. Now they've been able to develop a patterned one rather than a random assortment, allowing for a massively increased density of these reads. And this is how now a benchtop sequencer is hitting the read numbers that I'll explain in the coming slides. The next development that I've alluded to is the integrated DRAGEN bio IT platform. More on that to follow though. And the final point as well would be the single integrated cartridge. So again, for those familiar with previous technologies, there's no longer separate reagent containers for the different components during the run. You would insert your library onto this cartridge, which then gets loaded into the system and run in a very similar fashion to the iSeq. So zooming in just a bit more on the real positioning of the NextSeq, what I've given here is just a comparison of the MySeq to the NextSeq 550 to the NextSeq 1 and 2000. And in each case, I've indicated underneath the name the maximum capacity in gigabases that the system can generate. And alongside that, a very rough, approximate, estimated based on list price, cost per gigabase of sequencing. So these prices naturally, please, are subject to change pending exchange rate fluctuations. But it really demonstrates why the NextSeq 2000 would allow for much higher sequencing data for the same cost. So looking at the MySeq, you'd be able to churn out in the highest capacity run 15 gigabases of data at a cost point of around 2,500 Rand for each gigabase. The NextSeq 550 is a big jump up at 120 gigabases, but the cost is at around 850 Rand per gigabase. When you compare it to the NextSeq 1000, which has the same throughput at 120 gigabases, the cost has come down somewhat from 850 to 600. But the true jump is at the NextSeq 2000, where you can generate again 3 times the data, so 360 gigabases at max throughput, at a cost of, incidentally, 360 Rand per gigabase. Shown along the bottom, and this will be repeated, so please bear with me, but are the different applications that can be run at those different throughputs. So 15 gigabases on the MySeq, that would be sufficient for running sequencing of panels or small genomes. And that's still where the system will continue to see high use, especially in applications where you can't afford to have to batch samples to utilize a lower cost per run, or sorry, per gigabase rather. Once you hit 120 gigabases of data, you then unlock applications such as NIPT or enrichment panels and oncology panels. And now with the NextSeq 2000 with the P3 reagents, so previously these would not even have been recommended on the NextSeq 550, but they're now unlocked with the NextSeq 2000, or applications such as single-cell sequencing, trios, circulating tumor DNA, exomes, and RNA sequencing. We'll zoom in on the applications a bit later if you're unfamiliar with some of them. But what I really want as the summary for the slide is just for you to understand that the NextSeq 2000, I feel, really will allow for larger studies and new applications in current labs. And that's by reducing the cost per gigabase, which will allow for either a higher coverage or even a higher number of samples for the same cost. Compare these terms that I've been using, the P3 versus P2 flow cells. This is where the difference comes in between the two systems. The NextSeq 1000, the smaller one, is able to utilize the P2 flow cells, where the NextSeq 2000 can make use of both the P2, but also the P3 flow cells. Now, because it's Illumina, of course, they have no compromise when it comes to quality. The two systems both produce very similar quality scores as well. The read numbers, though, are where the major difference can be seen. You get 3 times the number of reads out of a P3 flow cell as you do from a P2 flow cell. That's 1.2 billion reads from a single run. And at 2 by 150, that's 300 base pairs per read, you generate 360 gigabases of data then in one run. And a third as much on the P2 flow cell because there's a third as many reads. Sequencing time naturally would be longer, around 2 days for a P3 flow cell, just over a day for a P2 flow cell, and at the costs that we described earlier. Now, if you, like the rest of us, have wondered why we have a P3 and a P2 flow cell, there will, hopefully before the end of this year, be the announcement of Illumina launching the P1 flow cells. These will be able to be run on both the NextSeq 1 and 2000, and this will fill the space where customers perhaps need to do a lower read number in a run, so where they can't afford to batch as many samples before a run. We don't yet have the scores for the qualities or the runtime or the cost points, but Illumina have confirmed that the P1 flow cell will have 100 million reads, which puts it, again, closer to a MySeq run perhaps. At 30 gigabases, that's the data of 2 MySeq runs. So really trying to just come down a little bit in the number of samples you would need to pool to do one run on the NextSeq. To put some numbers to all of these stats that I've been throwing around, I've highlighted here just 3 different applications that are really recommended on the NextSeq 1 and 2000s, and then along with certain assumptions, the number of samples that can be done. So for example, if you were doing single cell gene expression work and you were looking at 4,000 cells and your target was to produce 10,000 reads for each of those cells, then you could utilize the 100 cycle flow cells and you'd be able to run 10 samples on the P2 flow cell, which you could do on either instrument. Or if you had 30 samples and a higher need, you could do that in one run with the P3 flow cell on the NextSeq 2000. Looking at whole genome sequencing as an application with the assumption of a 50x mean target coverage, using a 200 cycle kit, you could do 16 versus 48 samples on a P2 versus P3 flow cell. And then finally, another featured application, shotgun metagenomics. If you wanted to allocate 50 million reads to each sample, then you could use the 300 cycle kits to really maximize the full data you get for every read. And then you could batch 8 samples versus 24 samples on the P2 versus P3 flow cells. So again, you'll notice it's always 3 times as much because there are 3 times as many reads on the P3 flow cell. This is a bit of a mirror from an earlier slide on the different applications. At the bottom, you'll see we have the MySeq being compared to the NextSeq 500, being compared to the current NextSeq 1 and 2000s. The 500 was later supplemented, with the NextSeq 550 to add array technology. But you can see that from 2011 to 2014 and now on to 2020, as these capacities have increased, we've also increased the applications that are recommended for these different systems. So you can definitely still perform your gene editing applications or targeted gene expression or amplicon sequencing on the MySeq, but you could do them in higher throughput on the larger systems, where you would then also, at the NextSeq level, unlock some single-cell capacity and metagenomic exposure, but really these shine on the NextSeq 1 and 2000 instruments, where the cost would be lower, and therefore your sample number could be higher in the same run. That's even on the P2 flow cells. On the P3, you could run 3 times the number of samples. And because of this increased sequencing depth, we can now have rugged trios, exomes, and deep metagenomics also coming into play. A rugged trio is a very interesting application whereby you would sequence someone with a rare undiagnosed genetic disease, typically these are children, as well as their 2 parents, hence trio. And then with a comparison of these 3 genomes, you would hopefully be able to diagnose their disease. But we have a case study on that coming up in a few slides. Similarly, we'll deep dive exomes and metagenomics as well. Before I do that, though, I really want to touch on this DRAGEN software that I've mentioned, and it was a bit of a thing to wrap my own head around it as well. But DRAGEN is both referring to the hardware that's required to run it, as well as a set of pipelines that were developed specific for that hardware. So DRAGEN stands for Dynamic Read Analysis for Genomics. And this combination was really developed to provide faster, more accurate, and more cost-efficient methods to run some of the favorite industry standard pipelines for genomic data analysis. And because of this, DRAGEN offers known pipelines, but at a higher efficiency and a higher speed. And this is great for both novice users, but also for expert users who are familiar with those pipelines already. Because DRAGEN, however, requires this dedicated hardware, you have to access it in one of 3 different ways. The first would be that you could have an on-premise DRAGEN server. So pictured on the right, you see there an example of what that server would look like. And you could house that and then feed it data from any Illumina sequencer. The next option would be to upload your data to the cloud and have it run via base space on a DRAGEN server that's located elsewhere. And the final option that I'm most excited about is that the NextSeq 1 and 2000 instruments contain an integrated DRAGEN hardware server already. So the hardware itself is physically attached to every single NextSeq 1 and 2000 instrument. Just to show you what I mean when I say hardware, this is an example of what it would look like. So we all know that computing can be done on CPUs. We're familiar with CPUs, and then also on GPUs, which have for long been a hallmark of the bioinformatics space. A similar system is an FPGA, a field programmable gate array. So the DRAGEN servers utilize both these FPGAs to really high success, as well as a few CPUs. And they're really an ideal platform when you want to perform parallel computations and a defined set of work, because obviously CPUs, GPUs, and FPGAs each have their own strengths and weaknesses. But what's really been demonstrated with the DRAGEN systems is the power of these FPGAs, which allow for accelerated analysis speed, a higher accuracy, and for more parallel computations to be performed. And this is really what's unlocked then these DRAGEN pipelines to be developed for this hardware. And when I say faster, here are a couple of examples. With the DRAGEN Bio IT platform, you would be able to analyze 57 genomes per day, versus previously you'd be limited to around 2.5 genomes per day. This is for previous existing standard solutions. So we didn't just pick the slowest ones to discuss, that was the standard. Similarly, an S4 flow cell, which is the sequencing chip at highest capacity used on the NovaSeq, it generates 10 billion reads from one sequencing run. And the DRAGEN Bio IT platform can demultiplex that in just 2 hours. And finally, going from FastQ to your VCF files for a whole human genome at 30x coverage can be done as quickly as 25 minutes. Now, with these speeds have come 2 Guinness World Records for DRAGEN Bio IT platforms. The first one, which is explained in more depth on the next slide, was 19.5 hours that the Rady Children's Institute for Genomic Medicine was able to go from sequence data to a diagnosis for a genetic disease. That still is the world record for the fastest genetic diagnosis. And the second Guinness World Record is for the fastest analysis of 1,000 genomes. So 1,000 whole human genomes were analyzed in just 2 hours and 25 minutes in 2017. And that was performed by the Children's Hospital of Philadelphia. And interestingly enough, in the DRAGEN Bio IT platform in the cloud. So not an on-premise server at all. To just give some more detail on the example from the Rady's Children's Hospital, they were really trying to establish a pipeline for using whole genome sequencing from blood samples and to compare that to their current models. And they found that it was 20 times faster to produce a diagnosis in this way than with standard of care. And not only was it faster, but they found that of the 42 babies that received both whole genome sequencing and standard of care as a diagnosis method, 43% were diagnosed using whole genome sequencing, but only 10% would have had a diagnosis with standard of care alone. So that's 4 times as many, which is truly remarkable for these rare genetic diseases. Speaking a bit more to the DRAGEN pipelines that are available, these are just some of them. So I've put a snapshot on the screen here of the various pipelines that come with the NextSeq 1 and 2000 instruments version of DRAGEN. But if you're going on the cloud or if you're hosting the server externally, there are a whole host of other ones available as well. And these were specifically chosen because they focus on the powers of the NextSeq 2000 and its capacity. So this would be the DRAGEN pipeline for germline, enrichment, RNA, single-cell RNA, amplicon, or ORNA compression, where specifically you were able to compress your FastQ files up to 5 times without losing any data. And just as a further example of what DRAGEN does, because I understand it's a very strange concept, this pipeline together with a hardware solution, this is an example of the DRAGEN Germline pipeline. And what it does are the 4 blue blocks at the bottom. So you would feed in your FastQ and BCL files. The DRAGEN pipeline is then able to do mapping and aligning. Together with BAM data, it then also does position sorting and duplicate marking. That process involves matching reads up that are identical to one another and then ignoring the duplicates to speed up your downstream analysis. That's where they are perfectly identical. Excuse me. At this point, you can then export a BAM file and proceed to variant calling into your VCF files. So the DRAGEN pipelines are all versions of this kind of a workflow. Depending on the exact pipeline, it'll have other branches coming in with new data sets for comparing and for mapping to reference genomes, or they might be slightly simpler as well. Now I'm going to focus on a few of the recommended applications for the NextSeq 1 and 2000 instruments, the first of which is small whole genome sequencing. Typically, this would be performed in one of 3 major ways, the first of which is the novo microbial genome sequencing. De novo, of course, refers to an example where you have no reference genome. You have no idea really of what your genome should look like, and you're assembling this genome for the first time. Because of that, it's of the utmost importance that your raw read accuracy and read depth are both kept to a maximum. And an example of where this was used that's, I think, very relevant to all of us is the paper highlighted on the top right, where the genetic sequence of SARS-CoV-2 was first described. Of course, this assembly couldn't have had a reference because in the beginning, it wasn't known what the virus was. The next major one would be not a de novo assembly, but microbial whole genome resequencing. So here you would be sequencing a whole genome to compare it to a known reference. And here, because you're typically looking for very slight mutations or insertions and deletions, it's again critically important that your accuracy is as high as possible, because when an alignment software detects a change, it has to try and decide whether that change is a result of sequencing error or whether it is an inherent mutation of the genome. So here again, Illumina shines, and again, a study based on coronavirus, but one that's even closer to home, is where we described what's now known as the beta variant here in South Africa for the first time. So this would have been resequencing against a reference genome, but still sequencing the full genome. And the final example, which is a separate application on its own to an extent, would be sequencing of either isolates or primary samples. So this would be environmental samples. In the case of isolates, whole genome sequencing of DNA would be recommended. But for complex samples, you would be performing shotgun metagenomics on the full set. So we'll zoom in on that in the next application. What I've just got here on the slide for you now is a comparison of the different Illumina instruments, because all of them are recommended for small whole genome sequencing, but with different strengths. So at the lower end of the spectrum is the iSeq, the MiniSeq, and the MySeq, which are excellent for lower throughputs. But where we really want to focus for today is the NextSeq 2000, which can do up to 384 samples in a run, which is only limited by the number of indexes that are available. So truly powerful for high throughput studies, like we're seeing now with COVID surveillance. So to zoom in on shotgun metagenomics as a whole new application, shotgun metagenomics really aims to take what was previously being done in the field of metagenomics, either using TRFLPs or 16S or ITS genes, but not just to sequence certain amplicons, but rather to sequence the total DNA from a sample. So it aims to therefore better answer the questions of who is there and what are they doing. As an example, you could take an environmental sample, and once you've amplified all of that DNA, there are certain workflows that can help you identify entire pathways like nitrogen fixation from that sample without ever needing to know what the organisms were that those genes came from. The reason that this was also in the small whole genome application highlights as well is that often from these metagenomic samples, given especially the read depth that a NextSeq can generate, you have enough reads to assemble at least some of the genomes that were present in those samples pretty much to completion. Of course, metagenomics is highly applicable in the fields of agriculture, environmental analysis, the human microbiome, where it's been shown that microbes actually play a much bigger role than we'd ever thought possible in modulating human health on levels from obesity through to cancer. And finally, we can also use shotgun metagenomics for pathogen surveillance and identification, and to monitor antimicrobial resistance genes, which I'll focus on in the next few slides. Before I get there, though, just once again to blow the horn of DRAGEN, there is a DRAGEN pipeline available specifically developed for metagenomics. So you would start with your complex microbial sample. You would perform your extraction and library preparation, followed by sequencing. For the purpose of this conversation, definitely on a NextSeq. And from that, the DRAGEN metagenomics pipeline can not only establish quality control, but it would also do your taxonomic assignment, and finally your organism classification. And from there, you could proceed with the rest of your downstream analysis that you want to perform on the samples. Zoom in on the particular applications here, where small whole genome sequencing and shotgun metagenomics were used. This slide here is talking about antimicrobial resistance genes. And obviously, we are all aware that this is becoming more and more of a talking point globally because of the widespread use of antibiotics and in some cases the overuse of antibiotics, and the fear that this would lead via perhaps horizontal gene transfer to the evolution of superbugs in the environment, to those genes hopping into unintended targets and also then conferring on them antimicrobial resistance. So the problem would be that if you're doing traditional targeted and hypothesis-driven screening, or perhaps 16S as a search tool, you would miss these novel organisms where these mutations have occurred. But metagenomics, where you're sequencing the total DNA, that will allow you to overcome those issues. At the same time as well, we know that there are many, many microbes that can't be cultured using traditional methods. And again, this is no barrier to metagenomics. So the authors of these various papers were really recommending that this needs to become a part of an international surveillance project to keep track of antimicrobial resistance genes in diverse habitats. And to go to the next slide, there was a further study that's highlighted on the bottom left, where sewage was actually used to do this surveillance as well. And once again, they say that sewage is an ethically acceptable and economically feasible approach with which we can do this surveillance. And highlighted on the bottom right of the screen is a study that I'll be zooming in on the next screen. But what was used here was Illumina's respiratory virus oligo panel, which is an enrichment panel. Now, these approaches typically have one of two initial steps. The first would be amplification of certain key genes using primers. This particular one uses enrichment, which is a different technology whereby you don't amplify a specific target out of the sample. You rather fish it out using a probe that hybridizes to that sequence, and then you discard the non-hybridized sequences. So you don't amplify it out, you rather fish it out using enrichment. Now, the respiratory virus oligo panel from Illumina was specifically developed in the SARS-CoV-2 pandemic. And on the next screen here, you'll see that it's able to then detect 40 other common respiratory viruses alongside the full SARS-CoV-2 genome as well. So really excellent application to not only detect variants and to perform surveillance, but also to have complementary information about co-infections that these patients or populations may be having if you're performing the analysis on wastewater specifically. And highlighted on the right is just a comparison of the different systems that this was targeted for, their runtimes, together with how many samples. And again, we can see just the massive sample load that you can put through on an NextSeq 1 or 2000, where you are limited once again by the indexes at 384. Moving ahead to the next one, the next application we'll be discussing really briefly is Whole-Exome sequencing. So the slide here just demonstrates the differences between Whole-Exome and Whole-Genome sequencing. Of course, in a world where we all have unlimited budgets and access to NovaSeqs, everyone would do whole-genome sequencing on every sample that they wanted to. But realistically, most of us are bound by budget, and that's where Whole-Exome sequencing can still provide an incredible wealth of information at a much more affordable price point. And it's also easier on your servers. As you can see on the top right, Whole-Genome sequencing at 30x coverage will generate around 120 gigabases of data. If we discard the introns and look just at the exons, we're down in the middle of the screen to 8 gigabases, and that's at 100 times coverage. So that's with a really deep sequencing as well. Your typical workflow for Whole-Exome sequencing would look something like this, where you'd begin with an Illumina DNA prep with enrichment with Illumina exome panel. So again, this word enrichment referring to the fact that we're not amplifying out the exons, rather we are fishing them out of the total sample set with the enrichment protocol. Following that, we would then perform sequencing. There in the example is a NextSeq 2000, but utilizing P2 reagent for the shorter runtime. And then finally, analysis on DRAGEN. And at the bottom of the screen, we're just echoing again the number of samples you could do on a P2 versus a P3 flow cell, under the assumptions that you are doing a 50x Whole-Exome sequencing run and that you are utilizing the 200 cycle kits. You could then do 16 versus 48 samples on the 2 flow cells in a single run. And sorry, I'm just going to go back for a second just to show you that using the DRAGEN enrichment pipeline, which would be the featured one for exomes, you would be able to interpret those in 15 minutes per sample. That's what it would take on the onboard DRAGEN bio IT platform. Next highlighted application we'll touch on is single cell sequencing, a very hot topic in 2021, and I think it'll become even more important as we get more and more access to systems that have the capacity to do the work, both the isolation of the cells as well as sequencing at a high enough capacity. So your typical workflow here would begin with tissue preparation, which would be some form of mechanical or enzymatic disruption to suspend the cells in single fashion. Then you would isolate those cells from one another. More on that in the next slide. And then following that process and the library preparation, you would then perform your sequencing and finally, of course, your interpretation. So just as a quick segue from Illumina, we at Separations also have ISL 8 systems available from Takara, as well as various systems from Cell Inc., such as the upside pictured on the right, that specialize in isolating, your single cells for single cell sequencing. So please, if this is a workflow that you're interested in, please get in touch with us. We would love to demonstrate the power of these systems to you as well. Illumina, though, what I've taken on the top there is a snapshot of the recommendations for the number of reads that are recommended for different single cell sequencing protocols from various kit manufacturers. So for those who are maybe not quite aware, Illumina is naturally an open platform when it comes to library preparation. So as much as we hope that we have the best solutions for you from our Illumina or Takara portfolios, if there is a different platform that, a different company that produces a library preparation method that you prefer, you can use that according to the manufacturer's instructions and still run that on your NextSeq. So you can see that the number of reads that are recommended per cell, they fluctuate between about 15,000 as low as 5,000, all the way up to 2 million reads per cell. So the number of samples that you could run would vary quite widely, but on the bottom we can see that if you're doing 4,000 cells at 50,000 reads per cell, on the 100 cycle kits, you could do 2 versus 6 samples on the P2 and P3 flow cells respectively. To talk a bit more about the single cell sequencing, it's really only been available in the last few years because of the really high accuracy and specificity that next generation sequencing brings to the table. And that is really critical for analyzing single cells with their really low abundance of DNA and RNA. And I mean, they've now been used in publications, as I mentioned there, to describe not only DNA mutations and copy number variants at the DNA level, but also at the RNA level. There's many applications looking at differing gene expression or RNA splicing. This makes this kind of technology relevant in fields such as oncology, immunology, neurobiology, to name a few. And on the right-hand side, I've put 2 that I feel relevant quotes from the Illumina literature spotlight on single cell sequencing. The first one says that understanding single cancer cells at their individual level and as an ensemble is bound to advance our understanding of not only therapeutic resistance, but all facets of tumor biology. And that's especially because of the incredibly high mutation rates within these cells, and especially because we know that tumors are not homogeneous in the distribution of mutations and of behavior of the cells. Looking at a whole other field of microbial ecology, single-cell genome sequencing of individual archaeal and bacterial cells is a vital approach to decipher the genetic makeup of uncultured microorganisms. Because if you only have in a sample 1 or 2 instances of a certain cell, it would be very difficult to sequence it without culturing if we went for single-cell sequencing. On the slide here, I've just stolen a slide from Maria's presentation a few weeks ago on RNA sequencing, showing just the workflow that our Takara Smarter Ultra Low kit, in conjunction with the Nextera XT kit from Illumina, would achieve for sequencing RNA from single cells. If this is an application that you're interested in, again, please reach out to us. We're really happy to get involved in single cell projects. So please lean on our expertise in this area. But just to tie it all together over here, single cell sequencing really is being run on the transcriptome, genome, and epigenome levels currently. I've highlighted on the right-hand side 2 articles just to show this, where the first one actually performed triple omics on these single cells. And in the second one, they looked at genetic and functional diversity. So in both cases, oncology studies, where I think we're going to see a massive drive towards single cell sequencing in the coming years, and hopefully fueled by the NextSeq 1 and 2000 instruments. This, I'm sorry to say, is near the end of my presentation. We've discussed the applications I really wanted to focus for today. So just to give a bit of a spoiler for next week, Mariette will be discussing specifically, well, amongst other applications, but the TrueSight Oncology or TSO 500 panel, also human whole genome sequencing, and more on gene expression as well, centered around that beast on the right, the NovaSeq 6000 instrument. So please Do join us again next week for the next installments of the presentations. And just to tie everything together for my talk for today, please, I hope that you understand that the NextSeq 1 in 2000 really straddles the position as the absolute top benchtop sequencer with a capacity that far exceeds the previous generations of benchtop sequencers, but still doesn't yet quite reach the NovaSeq 6000, but at a very attractive price point per gig to hopefully unlock deeper studies at more affordable price points. And with that, I thank you.
Thank you, Pieter. Can I just ask the team if there's any questions in the chat, or if there's any questions that you may have, you can unmute yourself at this point and just ask Pieter the needed question.
Nothing in the chat.
It's always frightening because I don't know whether everyone just understood everything or whether everyone is making coffee in the background. Okay, I think then, oh, sorry, yes.
Will there be any practical training available?
On the NextSeq 2000 and the 1000.
It just says, hi, will any practical training be available?
Oh, sorry. Well, what we'd be able to do, sorry, when we install the systems, definitely yes. And I'm not sure to what extent I'm allowed to say this, but at some point soon, there would be a capacity, we believe and hope, for giving some exposure to these systems right here at separations, we hope, but sorry, I can't say much more than that at the time. So no, currently we can't just give access to a system yet, but no, definitely when we install these systems, they come with full training. Maria normally gets planted there for multiples of days to make sure that your protocols are all running as they should be from end to end. I hope that answers the question.
Can we get the recorded audios?
I think, Colette, the link will be sent out later on, correct, for the session?
Yes.
After the whole session, or sorry?
Sorry, the sessions will be made available after all 5 sessions have been completed at the end of next week.
Okay, thank you.
Peter,
Perfect.
there's another question.
I see it. Thanks from Renata. Hi, thanks, Renata. So yeah, the DRAGEN functionalities are a part of the server and they can be run on the server, but there is a licensing requirement to continue using DRAGEN. So the option would be that you could buy credits to run them on base space, or if you have the NextSeq 1 or 2000, you could extend the license on that instrument. Those price points can all be discussed. With the initial sale, a 6-month license is included already so that you can see whether those pipelines are something that you're using. And I think the specific reasoning here was that a lot of people already have established pipelines in place. So I don't think that everyone would see the value necessarily in the DRAGEN pipelines. But yeah, the licensing options are there, and that would then give you full access to them as you extend those periods. The cost we would quote for you along with it, sorry, I'm not familiar with the list pricing just off the top of my head. I know that there is a lower cost version to extend the first 6 months up to a year, but that'll all be included in the quotation for the instruments up front. And again, sorry, it would be comparable to using the tokens and doing it on base space as well. So in line with what I think similar pipelines would have cost in the past. But we can get in touch with you and then we can just discuss the pricing more specifically if that's okay. Thanks. Any other questions?
Doesn't look like it, Pieter. So thank you very much for, oh, there's one more from Fadila. Maybe you want to take that one?
Yes, no, I see it there. Perfect. Thanks. So in terms of the best recommendation, realistically, it's the real world. And I think perhaps to the disappointment of a lot of my applications colleagues, we would often have to recommend based on the budget primarily. We would always recommend to get the best bang for your buck. But that being said, even if you had a much higher budget available, we would still recommend a smaller instrument in the case where those would be your major focus applications. So for example, if all of your workflow would be to investigate viral genomes, and especially at a lower throughput, it wouldn't really make sense to aim for the higher end of the spectrum. But we would always try and converse with you and see what applications you intend to run in the system, especially throughout its lifetime. See if there's a space for perhaps collaborating with other users so that you can get access to a higher budget. But yeah, we would typically recommend based on your applications primarily. I hope that helps. And then thanks, Renata. I see about the maintenance requirements for the instruments and the laser life. Maria, sorry, again, I'm just the sales monkey here. We don't have our technicians on the call, but do you perhaps have some info on the lifespan, the maintenance of the lasers?
There is no maintenance on the instrument, daily maintenance or weekly or so, like we have with the other instruments, because it's a dry system. We don't have fluidics on the system. The fluidics is in the cartridge, so there's no washes that are required. But with the instrument purchase and purchase of the extended warranty, there is a preventative maintenance which we do every year. I'm not sure about shelf lives yet, but we could find out. I mean, the instrument is fairly new. It's only a year or so, well, almost 2 years on the market. So it's not something I don't think it's something we know yet, but we can find out what is there, like globally any issues and how long do those units last.
I think to echo that, thanks, Maria. I think to echo that, yeah, definitely we can't definitely say with certainty that this will last 10 years because there hasn't been one running for 10 years yet, but we're not aware of anything in the field at the moment that's been a critical error. And again, quite the contrary, the systems from Illumina, typically they would undersell the potential until they can consistently show the upper value. So to speak for the NextSeq 2000, if you look at some of the older literature, it says that the upper capacity is 330 gigabases, because only recently have they again and again shown that the upper limit is 1.2 billion reads, not 1.1 billion. And so they've now officially supported that as the upper limit for the systems. So I think it'll be a similar story for the instrument lifespan. We can confirm a certain minimum lifespan that we would back it for, but realistically, as it goes, it'll last the longer. But as Maria said, no need for rinses in daily maintenance or monthly maintenance with that all-inclusive cartridge. I hope that's enough detail or not. Again, we'll definitely set up a call if we can soon, and then discuss any more questions that you have.
Thanks, Pieter. Ladies and gentlemen, is there any further questions? If there is, please type them in the chat quickly. No pressure, as your fingers are trying to get the questions to Pieter. If nothing, I would like to thank you, Pieter, for sharing your knowledge with us this morning. I think we can all contest to the fact that you are an expert in the field. So, yeah, you are very shy, but yes, it's all very well done. And then, ladies and gentlemen, we will see you next time.
Thanks, Colette. So I just want to echo, I've been accused of being not an expert, but not ever of being shy. But thank you very much. And thanks everyone as well for attending. Please, if there are any other questions, feel free to email me. It's on the screen there, or just get in touch with us.
Thanks. See you next week. Same time, same place.
Perfect. Thanks, everyone.
Bye-bye.